So hang on 1 second here. All right. According to this, we are recording. Okay, so welcome to the visual system. The visual system's job is basically to take an external stimulus, light, and transduce, convert it into electrical signals that your brain can interpret. Okay, so I'm going to use the visual system to teach you some key concepts for all systems. Actually, hang on a second. I'm a little nervous that it's not actually recording here. Okay, we are recording. So this process is called transduction. All transduction is converting a signal from one form to another. Okay, so what we're going to actually do is we'll take light information, we'll cut it in, or we'll transduce it, we'll convert it into electrical signals, which will then be perceived by the cortex, the cerebral cortex. Okay, so this is the job of the visual system. Now, when we talk about light information, there's obviously a lot of information there. Here we have a pretty picture of a vertebrate retina. The retina has blood vessels throughout most, but not all of it. Here we have something called the optic disc. This is where axons leave the retina. Okay, so axons will leave the retina through that optic disc. Here we have the macula. And within the macula, which is that whole thing there, we have the fovea, which is this spot. The macula is an area with no blood vessels. Yeah, I drew it a little big. The fovea is the center. And this is where we have sort of our best vision. Okay, so the stuff I am going to go over in more detail as we move along. You can actually see the structures in an actual retina. All right, so for vision, I want you to think in terms of what we need. We need a stimulus. What's the stimulus? I just told you, light. We need a receptor to detect the stimulus. If you don't have a receptor for it, you can't detect it. So think about magnetic fields. Humans can't detect magnetic fields. How come? We don't have the right receptors. Okay. So in the case of the visual system, anything having to do with light is called photo, right? So here we'll be talking about photoreceptors. Not very sensible name for it. So photoreceptors will be used to detect the stimulus. Once we do that, we need to transduce that signal into electrical signals. Initially, we're going to use graded electrical signals. So we have talked about action potentials. We'll use action potentials But immediately in the photoreceptors, we're going to have EPSPs and IPSPs. These are graded potentials. They can be a little or a lot. And then we need a processor to make sense of the electrical signal. So these graded electrical signals will get get converted into action potentials. Those action potentials will then get to our processor. And when we talk about any sense, the cerebral cortex is going to be that processor. A lot of your cerebral cortex is devoted to vision. When you look at other senses, we devote a lot less cortex to it. Vision takes up the most sort of brain space of all of them. Now here, when I talked about these, I am talking about vision, but this is true of all senses. So in order to hear something, right, we'll have sound waves, pressure waves in the air. We need a receptor to detect that stimulus. We need to turn that into graded potentials and then a processor to make sense. So this puts all your senses in context. So when you are studying the other senses, think in these terms. Okay, what's the stimulus? What is the receptor to detect that stimulus? How is it transduced into electrical signals? Where is it processed? If you can answer those basic questions for all the senses, you'll be in fantastic shape. Now, light. Light is part of the electromagnetic spectrum. If you look at the electromagnetic spectrum, it is includes things like gamma rays, X-rays. These are cosmic rays, ultraviolet, microwaves, TV radios. All of these are part of the electromagnetic spectrum. Humans can see 410 to 770 nanometers. I want you to remember that. This is visible light, 410 to 770. 410 is going to be, I want you to remember, is in the blue range. 770 is red. Okay, so short wavelengths, about 410, blue. Longer wavelengths, 770, red. I want you to remember this. Now, this is all that's visible to us. Outside of this, we can't see it directly. But as you know, right, we have detectors that can find UV light or infrared light. And we can use these to convert electromagnetic energy outside our visible spectrum into things that we can see. All right, but we have to use a special sensor. This class is very much focused on human. That's good because that's what you're interested in. I'm a comparative guy. I like thinking about all animal species. When we think about the visible light for a human, it's much smaller than the range for some other species. So insects, for example, honeybees in particular I'm thinking of, can detect ultraviolet light. So when we look at a flower, ooh, it's pretty, all that stuff. When a bee sees a flower, it sees a completely different pattern. Okay, so one thing I want you to remember is this is what goes on in humans, but other species have senses well beyond ours. All right, so light's electromagnetic energy. The color of an object is dependent on the color of light it reflects. So if an object is orange, what that means is that it is going to absorb light from all these others. The light in the orange range is going to bounce off that object and hit your eye. Remember, the light has to get to your retina. That's what you perceive. If orange were being absorbed, that light would never get to your eye and you wouldn't see it. Now, the way our sensory system is worked out too, it's the wavelength that determines color. If we talk about visible light, white light, again, this is a question you don't have to answer out loud, but what spectrum, what wavelengths are in white light? Hopefully you remember all. So if we have all the wavelengths together, all the light together, we get white. How about black? Black is what we perceive when there are none. Okay, so one thing, the book talks a little bit about this. I'm not going to go into detail about it. We're talking about light, not crayons. Okay, so if you take crayons or paint and I mix blue and green and yellow and orange and red, what color am I going to get? If I mix pigments, I end up getting eventually ugly color, but eventually we get black. Okay, if we mix light, if I mix blue, green, yellow, orange light, I end up getting white. So remember, we're talking about light, not crayons. Sometimes this mixes students up. All right, here's just another way of showing it. Again, the electromagnetic range, 410, blue. You'll remember that, 410. 770 is red. Okay, so that's what we can see. So that means that the photoreceptors, the photopigments that we have, are going to respond to wavelengths within this. Outside of this range, the photoreceptors won't really respond. One moment of disclosure: when it comes to ultraviolet, it turns out that we do have short wavelength photopigment. They used to be called the blue cones. Those can actually respond a little bit to ultraviolet light. The lens of the eye, though, absorbs UV light. So if there is UV light around, it gets absorbed by the lens and it never actually makes it to the cones. So humans don't have an opportunity to respond to ultraviolet. But anything in the infrared, for example, or too large in the ultraviolet range, I'm sorry, too small in the ultraviolet range, those we don't have photopigments that respond to. And just real quickly, remember when we talk about an object and we're talking about color, our perception of color depends on what light is reflected and what light is absorbed. Okay, so again, an object is going to, when you shine light on it, it will reflect. It will reflect some wavelengths. I'm just going to write lambda. Lambda equals wavelength. Okay, so some wavelengths will be reflected. That will allow it to continue on here and hit my eye. Others will be absorbed. These won't hit the eye. All right, so those are the basics of the stimulus. Let me pause here for 1 second. Okay, so that's the stimulus. Now we're going to talk about how we detect the stimulus. This is a cartoon of an eye. I'm not going to go through iris, cornea, lens, all that stuff. If you don't know it just from life, you can read about that in your book. When we have an object, what's going to end up happening is light's going to strike that object. It's going to be reflected off the object. That is going to enter into our eye and hit our retina. Retina is where the photoreceptors are. When we stimulate the photoreceptors, we're going to get some graded potentials. Those potentials are going to be converted into action potentials. Those action potentials will be generated by cells called ganglion cells. I'll go into this in more detail and give you an overview, so don't panic. Those ganglion cells will send axons into the brain. They'll first go to the thalamus, then they will go to the cerebral cortex, where processing will occur. It's a fairly complex system with lots of connections, so I'm going to walk you through it slowly. If we look at an object and the light from that object passes through a pinhole, your pupil is basically a pinhole. When things pass through a pinhole because of physics, this is pure physics, the image is inverted and reversed. So if we have light from the top of the pen, as it passes through, it's actually going to get inverted and it will hit the lower part, the bottom of the retina. If we look at the bottom of the pen here, that will then be the top. It's not shown here, but right and left also get reversed. So when it hits the retina, left and right. Now this doesn't matter because your brain compensates for this change in the image, but you just need to understand this. Because when we move to the connections, it will mess you up if you don't remember that. All right, so we're going to strike the retina. The retina has the photoreceptors, which are the rods and the cones. Remember I talked about the macula, right? So here is a cartoon of the macula. Here we have a little tiny pit, that's the foveal pit, that's going to have some adaptations to make sort of our best vision. One thing you'll notice here, I talked about the optic disc, that's the physical basis of your blind spot. If you look here, we don't have any photoreceptors. We have all the axons leaving the brain. So if the light hits the blind spot, you wouldn't detect this part of it. Now your eye is constantly moving so that the image doesn't stay on the blind spot and your brain sort of fills in the blanks. We'll talk about that a little bit more. All right, here's a close-up. So remember, light's coming this way. Here we have the photoreceptors. Okay, photoreceptors. These are going to be connected to cells called bipolar cells. And these will be connected to cells called ganglion cells. They're really, strictly speaking, called retinal ganglion cells. I'm going to use shorthand and just call them ganglion cells because it's obvious we're talking about the retina. But properly, make sure you use retinal ganglion cells. All right. These retinal ganglion cells will then send axons to the brain. So here we're going to the brain. All of those are going to bundle together and form the optic nerve. You'll also notice in this bundle we have blood vessels coming in. So the retina is actually a vascular tissue. You need to feed it or it will die. Okay, one thing I want you to notice, where is light detected? Light is detected by this part of the photoreceptors. So if I have light coming in from the outside world, you see a problem here? I do. This is the dumbest thing I've ever seen. If I want to detect light, light has to pass through axons. It's got to pass through ganglion cells. It's got to pass through connections here, all through my bipolar cells, through the cell bodies. And now finally, I get to the photoreceptors. So humans have what is called an inverted retina. Photoreceptors are at the back. This is stupid. If you were an engineer and you designed a light sensor where it had to pass through your circuits to get to this, you'd be fired, obviously. Now, there are issues with light going through. As it goes through, light refracts. So when it gets into the fat, it's actually going to change the pathway a little bit. As we go through different densities, it scatters the light a little bit. So it's not as precise as it could be. Okay, so there are consequences to having to pass through all of this. Why is it like this? Because that's how it evolved. Not a very satisfying answer, but that's why it is like this. I'm going to talk in a minute. It doesn't have to be this way. There's something called averted retina, where the photoreceptors are on the outside. You see this in so-called primitive animals like cephalopods, octopus and squid. Their retina makes sense. Ours does not. Okay, so the other consequence of having this arrangement now is the photoreceptors will detect the light, will switch it to electrical and chemical signals, get that to these ganglion cells. Now, these ganglion cells have to send that information to the brain. In order to do that and leave from the inside of the retina to the brain, we are going to basically have to create a space where the axons leave. In a verted retina, we don't have to do that. They're already down here. So the blind spot is a consequence of having an inverted retina. All right. This is just a cool picture of an actual retina. When you see a histology, a picture of a retina, it's usually photoreceptors at the top and the ganglion cells at the bottom. All right, so here I just want to show you in this picture, this is what it actually looks like if we run some histology on it. This is a low power electron photomicrograph. These are all going to be cell bodies. Okay, so you can see these are organized in terms of layers, where we have a layer with lots of cell bodies, we have fibers, we have more layers. There's more complex names for all of these. We're not going to go through it. ONL stands for outer nuclear layer. So this has the photoreceptors. Just know there's a photoreceptor layer. Next, we have the so-called internuclear layer. This is where the bipolar cells live. And then we have the ganglion cell layer. This is where the ganglion cell layer is. Okay, so basically, remember, we're going to go from photoreceptor to bipolar to ganglion to brain. Okay, that's the flow of information. This is looking at the human fovea. Okay, so with the human fovea, there's, sorry, with the human fovea, there's a couple things to keep, to keep in mind. It's the place where we have the best vision. So when you read, you move your eyes, right? The reason you move your eyes is because you are trying to get the image onto the fovea, right? So you probably already know rods and cones. I'll talk about these in more detail. Rods, low light, very sensitive, but low resolution. Cones need bright light, high resolution. Okay, so we use our rods sort of at night. When we're doing something like reading, we use our cones. So one of the things to notice about the fovea is that we only have cones. Okay, so remember I showed you the nice layers. I've got my photoreceptor layer, I've got my bipolar cells, and then I have my ganglion. If we look here at the fovea, it doesn't look like that at all, does it? What's actually happened is the cells that are in the ganglion cell, the bipolar cell layer, the photoreceptor, have been pushed to the side. Okay, so they've been squeezed to the side. So if I'm light, remember now light's coming from the bottom. Here's light. I got to pass through all this stuff here before I get to the photoreceptor. All kinds of opportunity to be diverted, refracted, scattered, all kinds of bad things. Here, I have a much more direct path right to the photoreceptors. So by pushing all those cells, all those axons, all that stuff aside, I create a direct path to the photoreceptors. Now, you can't do this indefinitely. Obviously, they have to go somewhere. So we have a small specialized region called the foveal pit where we have direct access to the photoreceptors. So this has functional and perceptual consequences. Other species have much more fovea than we do. We only have a fairly small amount of fovea. The problem with the fovea, again, humans are not the best at everything. We're not the worst at everything, but we're pretty good at everything. So humans are kind of a compromise. If we were mostly rods, we'd be really sensitive at night. We'd be able to see better. You know, night's a scary time for us because we can't see anything. If we had more rods, we'd be better at night. But without cones, we wouldn't have high resolution vision. If we were all cones, we would be completely blind. One way to kind of find your fovea and the function of it is if you go out, it's harder in Singapore, but if you go out and you find a faint star, like sometimes you'll be looking up at the sky and you'll see a faint star. You look at it and it disappears. Then you move your vision just to the side of it and it comes back. And then obviously you're tempted to look directly at it because you want to get a better view of it, right? And you think if you look directly at it, you'll be able to see it better. Basically, what you're doing is when you first see it, it's in your peripheral, the light's hitting the periphery of your retina. Lots of rods, very sensitive, and you can detect it. When you shift your eye to look directly at it, what you're trying to do is put it onto your fovea. When we put it onto the fovea, the problem with that though, only cones. And with a faint star, they're not very sensitive. So by trying to get a better view of it, we do put the light successfully on the fovea, but the cones aren't sensitive enough to pick it up and you don't perceive it. Okay, so again, just functional aspect of it, and we'll talk more about this as we go on, but these are the basics. One thing I just wanted to point out with ours, I talked about an inverted retina. This is a so-called verted retina. This is how the retina works in a cephalopod. Here you look, we've got our photoreceptors. So here comes light through the lens. It's going to go and it'll strike here. This, what it's striking, is the photoreceptor. So it hits the photoreceptor first. Then that information is transferred to bipolar cells, ganglion cells, and goes through to the brain. Now, if you look at this, no blind spot. So a cephalopod doesn't have a blind spot. Okay, so again, this suggests two things. Evolutionarily, eyes have evolved more than once, at least 3 times, because insect eyes are completely different. But it also suggests that cephalopods actually have very advanced vision. All right. So now I'm going to spend some time focusing on rods and cones. So this is a sort of a Photoshop doctored up scanning electron photomicrograph of photoreceptors. Normally it's black and white, but they've added color to it. So here we can see this photoreceptor here in yellow and this photoreceptor in blue. And if you look at the shape, they're different. This is kind of a, I don't know, rod shape. And this seems kind of cone shaped. So this is why they're called rods and cones, just simply by the shape. Within cones, we have 3 types. Okay, they used to just be called red, blue, green. We don't use that anymore. I'll go over that in a slide in a minute. These are the physical basis where we take light to electrical signals. So this is where the action is. If we look at the rods and cones, they are sensitive to different wavelengths of light. So here on the y-axis, we have percentage of maximum response. And here we have the different types of photoreceptors. I'm going to start with rods. Okay, so if we look at the rods, that's this curve. Rods, if we have a wavelength, we shine 400 nanometer wavelength, a rod will have a small response. As we move towards, say, 500, so 500 nanometers, now we get our maximal response. Okay, so all photoreceptors, including rods, have wavelengths where they have their peak response. Now you'll notice that none of these respond just to one wavelength of light. Right? They respond to a range. But if we get away from the peak response, the wavelength at which we get the peak response, we get this decline. All right, so rods are here. If we you can convert it to blue, green, red if you have to, but now the preferred nomenclature is short, middle, long. So here, if we look, our blue or our short wavelength, you know, around 410 nanometers or so. Okay, so if we look, these were done biochemically. These weren't done measuring in the eye itself. Okay, we don't have the technology to do that in a living organism. So remember what I told you about the short wavelength, how they do kind of respond to UV ranges, but because the lens blocks it, it never makes it. All right. But you can see if we look at the peak here, we look at the peak here, and we look at the peak here. Let's say I shine 500 nanometer wavelength light. Which cone will respond? Well, my short wavelength, small response. How about my long wavelength? They'll respond, sort of a middle respond. How about my middle wavelength cone? It'll respond even more. All right, so if we look at 500 nanometers, I'm just going to poorly draw that line up. If we look, we have a response profile that when we look at all three of the different types of cones, we have a unique response profile. Okay, so I'm going to jump ahead a little bit real briefly because this is going to apply for perception of color. Let's say my red or long wavelength cone is responding at 75% of its max value. What wavelength light do I have? Well, let's draw this over. 75, we got this. Oops, it could be this. So if all I know, if I have one cone, I can't differentiate between something at say 525 and something at say 615, right? Both of those I'll have 75%. So if my long wavelength cones are responding at 75%, my brain doesn't know whether it's 525 or 615. Okay, I can't tell the difference. So just one photopigment doesn't let me distinguish amongst wavelengths of light. Now, what if I added the rest of the cones though? And so I know that I've got a ratio of activation. So my short equals 25, middle equals 70, my long equals 50. Okay, so there, if I look, there's only one place where that is possible. Okay, so more photopigments equals better resolution of wavelength. And that lets us distinguish colors better. So the more you have, the better you can do. So if you only have one, you won't be able to distinguish wavelengths, and color vision isn't the case. If you've got two, you'll be able to do it better. Three, even better. Humans, as you know, we have three. Other species, some goldfish have 12 photopigments. I think it's 12, something like that. They have way more photopigments than we do, so they are much better at distinguishing colors. Okay, I'm going to step away from color vision though and talk about rods for a second. I'm going to talk about the chemical process. So what I'm actually going to do is pause for a second. All right, so now talking about the rods are going to be the photoreceptors we use in so-called, let me change my color here. Okay, so this is low light. So the rods have some characteristics. They're highly sensitive to light. And so the transduction is very, very efficient. they're easily bleached. What I mean by bleached is we can run out of photopigment. What happens if you run out of photopigment? If we run out of photopigment, we can't respond anymore. And by respond, really, I mean detect. So if you go out on a very bright day, for example, and you can actually get to the point where we have no photopigment left in your rods. Now, you're not totally blind because you still have your cones, but this can lead to issues. You don't have it in Singapore, but we have snow blindness in the Arctic, where snow is white, which means it's reflecting all the light. So it's very, very, very bright. The light comes, the light shines, hits your rods, uses up all the photopigment because there's so much light. When you use up that photopigment now, suddenly, even though there's still light there, the thing you need to detect it is gone and you can't see anymore. So rods easily bleached. These are concentrated in the periphery of the human retina. You don't have any in your fovea. So again, I told you about this star, right? You might see it in your peripheral vision. The moment you look at it, it's gone. That's because of the arrangement of rods and cones. These have no color information. Okay, so these black and white vision, if you think about it and go home and try it. If you turn down the lights and you look at a color picture, obviously not a screen, you need a physical picture. If you turn down the lights, you'll get to a point where you can still see the image okay, but the color is all gone. That's where you've reduced the intensity of light to below what cones need to operate, but it's still enough that the rods operate. So you can actually see that yourself. Now, this is the chemical reaction. So the book talks a little bit about it. I'm going to go through this in several different pictures. I definitely want you to know this. So rhodopsin is the photopigment in rods. Okay, so this is a complex chemical. It is going to be more than just one component. So if you look down here at this picture, this is rhodopsin. Okay, so it's going to be a chemical 11-cis-retinol plus this big protein called opsin. Okay, together this is rhodopsin. Okay, so 11-cis-retinol, it's written, and opsin. These have to work together. If you don't have this, it won't work. If you don't have this, it won't work. All right. When light strikes this complex, it induces a change. It's basically going to shift that 11-cis-retinol, which is shaped like this, to all trans-retinol, which is shaped differently. Cis and trans, these are just chemical terms about the conformation of it. You don't have to really know what they mean other than that it's about the chemical structure. Now, when we straighten this out, when light hits it, and through a series of events makes this all trans-retinol, it changes the shape. When it changes the shape, right, it's going to interact with the opsin and change the shape of that. And it's actually going to allow these two things to separate. Okay, so light is going to take rhodopsin and it's going to cause it to split into opsin and all trans retinol. Okay, so here we are ready to go. We're exposed to some light. Very quickly, we get this change into opsin and retinol. Okay, so remember how I said they're easily bleached? Each time light strikes, it's going to destroy the pigment. It makes it inoperable. It doesn't work anymore. Now, if we want to see again, enzymes, energy, vitamins like vitamin A are going to take these two components and put them back into the rhodopsin and have them ready to go. You can see the word fast and slow here. So light does this really quickly. So we break it down real quickly. We rebuild it more slowly. You know, we've got tons of this hanging around. So if we're exposed to, you know, moderate levels of light, we can draw on our supply. So even though we're rebuilding it more slowly than we're using it, we still have enough to overcome that. But there are occasions where there's so much light, so much splitting now that we get this conversion into the inactive forms, and we can't build it fast enough. Okay, so this is the nuts and bolts of the transduction process. Now, when we get this conversion, this is going to have effects on the photoreceptor, on the biochemistry of the photoreceptor. Okay, so I'm going to go through that in a minute here. Now, photoreceptors are nutty. I'm going to spend some time going through this. It's going to take us a while. We're running short on time and all of that. I don't care. This is confusing, but you need to know the details of this. And it's nutty. This is counterintuitive. Okay, so the visual system started out stupid, having light to go through all those cell layers to get to the photoreceptors. Now it gets wacky. So I'm going to go through and try to explain this the best I can. So here we have a rod cell. The rod has two components. We have an outer segment and an inner segment. Okay, the inner segment is just a regular cell. I got a nucleus. We'll have mitochondria, protein synthesis, all that stuff going on. Okay, so this is just a regular cell. Part of it is kind of pinched off a little bit, and this is called the outer segment. This is where the photopigment is. Okay, don't worry about memorizing that. That's just to help you make sense of the diagrams that are coming up. So transduction, light transduction occurs in this outer segment. What goes on here is going to affect the membrane potential of the rest of the cell though. Okay, so here's something I'm going to tell you about rods and cones that will hopefully help make sense. The membrane potential of these is about minus 30 to 40 millivolts. Okay, so we learned minus 70. In rods and cones, it's a little different. It's about minus 40 millivolts. Sorry, I screwed that up. Minus 40 millivolts. Okay. When the cell is depolarized, it releases neurotransmitter. And that neurotransmitter is glutamate. These cells do not generate action potentials. Okay? The amount of glutamate released is proportional to the membrane potential. So more negative, hyperpolarized, depolarized equals more glutamate being released. Okay. Now, here's kind of the crazy thing. Normally, we would think that if a stimulus was present, the cell would release more neurotransmitter, right? That's the intuitive way for things to work. So something's touching me, those cells that are going to detect that will get excited and fire a bunch of action potentials. That makes sense. That is not the way photoreceptors work, though. Photoreceptors, again, are really wacky. Okay, so remember these. Keep these in mind about these nutty, wacky, counterintuitive cells. When we are hanging out in the dark, okay, our membrane potential here is going to be about -40. Okay. The way the situation goes, remember, there's no stimulus here. So I've got my rhodopsin, okay, the photopigment just hanging out here. This is connected to a G protein. Okay, without light, these G proteins here are inactive. They're not doing anything. Normally in the photoreceptor, second messenger, this is a second messenger, levels are high. This cyclic GMP, this second messenger, is going to hold some channels open. And those channels are a sodium and a potassium channel. So normally at rest with no light around, sodium channels and potassium channels are open. That means sodium's going to come in and potassium's going to leave. Now remember from the action potential, when we have sodium in and potassium out flowing, who wins? Sodium wins. So as sodium comes in, that's going to constantly depolarize the cell. Okay, so at rest, in the dark, no light around, my rhodopsin is hanging out, my sodium channels are open, sodium comes in, and that's going to lead to a slightly more negative membrane potential than we're used to. Now, with this -40, that's a depolarization. That is going to cause the release of neurotransmitter. Okay, so remember, we're going to go from photoreceptor to bipolar cell to ganglion cell. Okay, so information is going to come from photoreceptor transferred to the bipolar. This is going to be through a chemical synapse. Okay, so the photoreceptor is going to release neurotransmitter that's going to do something to the bipolar cell. This bipolar cell then is going to send connections to a ganglion cell. And then that is going to go on to the brain. That's going to go on to the brain. So we have neurotransmitter being released here. Normally, again, the sensible thing, the intuitive thing would be that light would somehow increase the amount of neurotransmitter. That would tell us there's a stimulus there, right? But again, that's not how it works. What is going on is in the dark, we release glutamate. Okay. And again, I just told you why. Because these sodium channels are open in the dark. That's going to lead to a depolarization. That depolarization leads to the release of neurotransmitter. Okay, so in the dark, the photoreceptor is releasing glutamate onto the bipolar cells. In the light, okay, we hit, right? We've got our retinol, our opsin, right? Things split. We go from 11 cis to all trans. What that is going to do is basically activate that G protein. It's going to change the amount of cyclic GMP around. You don't need to know the details of this. Let me simplify it for you. Okay, so there are some biochemical reactions that go on. You don't need to know those. Light hits, separates my photopigment. That is going to cause those sodium channels to close. Okay, so that's what I want you to know. Light hits the photopigment. That's going to result in the closing of those sodium channels. Now, when those sodium channels are closed, sodium can't flow in anymore. So if sodium can't be flowing in constantly, now my membrane is going to hyperpolarize. I told you that the amount of neurotransmitter released is proportional to the membrane potential. When we're hyperpolarized, that means we're going to get less release of neurotransmitter. Okay, so in the light, we actually get decreased glutamate being released here. I know that makes no sense, but that's the way it is. Okay, so counterintuitively, light reduces the amount of neurotransmitter released by the photoreceptors. We know in great detail the mechanism by which it occurs. Again, the point of this, don't panic when you see this, but it goes through the steps again. I'm going to go through this a few times. Hopefully it'll help you remember. So when light hits our photopigment, right, it's going to change the shape of the retinol. It's going to cause it to split with the opsin. When that happens, we get a second messenger system that's activated. Again, we know the details of this in great detail. That is going to result in the closing of the sodium channels that are normally open. That means sodium can't flow in anymore. And that is ultimately going to lead to a hyperpolarization. This is a diagram from your book. I'm going to go through this again. I know it kind of gets old hearing it, but this is confusing and something that you need to know. So in darkness, the photopigment is rhodopsin. That's a retinol 11 cis plus opsin together. Normally, the photoreceptor cell is going to produce a second messenger called cyclic GMP. That second messenger is going to cause sodium channels to open and remain open. That depolarizes the cell. When the cell is depolarized, it's going to release neurotransmitter. The neurotransmitter of the photoreceptor is glutamate. And so this is what goes on when there's no stimulus around. When there is a stimulus around, the retinol is going to absorb the photon. That converts the 11 cis retinol to all trans retinol. The rhodopsin molecule breaks apart. When it breaks apart, it's basically going to cause enzymes to be activated that are going to break down that cyclic GMP, that second messenger. Remember, the job of the second messenger here is to hold those sodium channels open. Okay, so cyclic GMP opens those sodium channels. Okay, so when those are, when there's plenty of cyclic GMP around, those channels are open. If we break down cyclic GMP, we no longer hold the channels open. Okay, so they close. When that influx of sodium is prevented, the photoreceptor becomes hyperpolarized. That means less glutamate's released. Now here's a little additional information I haven't mentioned yet. The more light is absorbed, the greater the hyperpolarization. So if there is more light, the greater the inhibition of the cell will be. The greater the inhibition of the cell, the less glutamate there is. All right, I'm going to pause here for a second. Sorry, I lost my pen. All right, so the photoreceptors. Photoreceptors are always active. Okay, so this is in the dark. Their default is to be active. And when I say active, I mean releasing glutamate. So they're depolarized and releasing glutamate. When light hits them, they're hyperpolarized, so they slow the rate of glutamate release. Now here's something that is kind of crazy again. Their synapse onto the bipolar cell is inhibitory. So here's my photoreceptor releasing glutamate. Here's my bipolar cell. Okay, so glutamate's being released. Now, what effect does glutamate have on the bipolar cell? One thing I want you to know, hopefully the first thing you're thinking with glutamate, hey, glutamate is the major excitatory neurotransmitter in the brain. What determines what a neurotransmitter does? So there's a little phrase we use constantly in bio. Receptor confers biological specificity. And what that means is basically it's the receptor that determines what a neurotransmitter does. For the most part, when we talk about glutamate in the brain, we're going to be talking about NMDA receptors, AMPA receptors. We're going to come back and visit those. Those are basically ion channels. The AMPA receptor is a sodium channel. So when glutamate binds to AMPA, it opens up that sodium channel. Sodium rushes into the cell and causes a depolarization. Here on the bipolar cells, though, we have a different receptor for glutamate. And it turns out that this receptor is inhibitory. Okay, so when glutamate is being released in the dark, I'm going to get lots of inhibition of the bipolar cell. So increased glutamate equals hyperpolarization in the bipolar cell. Right? I told you the visual system was nutty. All these exceptions, it's crazy. Okay, so photoreceptor in the dark is going to be depolarized, releasing lots of glutamate. That glutamate is going to bind to and activate receptors on the bipolar cell. When those receptors are activated, we get a hyperpolarization in the bipolar cell. Now, just like the photoreceptor, neurotransmitter release in the bipolar cell is proportional to its membrane potential. So when the bipolar cell is hyperpolarized, what happens to neurotransmitter release? It goes down. Now, when light hits the photoreceptor, the release of glutamate decreases. If I have less glutamate, what does that do to the bipolar cell? I have less inhibition. Less inhibition leads to release of neurotransmitter. Okay, so without that inhibition, the bipolar cell is actually depolarized. It's excited, and I get more neurotransmitter being released. Why the system is like this, I wish I knew. It has to do with the evolution, obviously, of the cell. All right. So this is a diagram that sort of puts things together for you. I like this as a good summary diagram. One thing I want you to know, photoreceptor and bipolar cells, no action potentials. These just have EPs, SPs, and IPSPs. The ganglion cell, this cell, finally, we're back to normal. So the ganglion cell behaves like the neurons you're used to. It's a photoreceptor and bipolar cells that are kind of nutty. All right. So remember, if we're in a human being, we've got our lens, right? Light comes through, passes through all this crud, gets through all these layers, finally hits the photopigment here in this outer segment of the photoreceptor. Now, when we are normally at rest, we're at minus 40 millivolts or so. Sodium's flowing in. Here I shine some light. What does light do to my membrane potential? Well, remember, light equals close sodium channels. And that is going to result in a hyperpolarization. Okay, so here we are, we shine light, we get a nice hyperpolarization in my photoreceptor. Now, what does this hyperpolarization do to neurotransmitter release? We decrease glutamate release. Now, at this synapse, this connection between the photoreceptor and the bipolar cell, remember, glutamate equals hyperpolarization. Okay, so this is just showing the stimulus given, which is just a flash of light. These were done with electrical recordings, so you can actually show this. And presumably at the same time, glutamate release is decreased. Now, if light is going to result in decreased release of glutamate from the photoreceptor, that means I have less inhibition. Less inhibition equals depolarization or excitement. That is going to increase the neurotransmitter release. That increased neurotransmitter here, we have an excitatory, regular old excitatory stimulus, synapse. So now if I increase neurotransmitter here, that's going to excite the ganglion cell. When we get excitation here, Now at the axon hillock, we get action potentials. Okay, so I'm going to walk you through light hits, strikes of photopigments. That is going to lead to a hyperpolarization. That hyperpolarization causes a decrease in the release of glutamate at the synapse. Since glutamate is inhibitory, if I decrease the release of glutamate, that means I'm going to decrease inhibition. Decreased inhibition is opposite, other side of the coin of excitation. That's going to cause this bipolar cell to be depolarized and release more neurotransmitter. When I release lots of neurotransmitter here, it's excitatory synapse so that this cell becomes depolarized. I'll exceed threshold at the axon hillock and action potentials will be generated. Okay, so that's kind of it, start to finish. For your cones, in terms of the details of the phototransduction pathway, things like that, focus on the rods. The cones are similar. Cones are relatively insensitive to light, though. They're concentrated near the fovea, and these are essential for color vision. No cones, no color vision. We talked about three, long wave, medium wave, and short wave. These used to be known as red, green, and blue. The field is switching, so instead of talking about red, green, blue cones, long, medium, short wave. I go back and forth. I was trained on both, so I kind of go back and forth. All right, so here I want to talk again one last bit on this before I stop. When we look at each cone, okay, so here we've got a cone with short. Its peak absorbance, that means where it is going to absorb, the photopigment absorbs the most light, is at 420 nanometers. Okay, so when I absorb the peak light, I absorb it all, I'm going to be able to have a strong response. Let's say I have 450 nanometers though. If we look, 450 nanometers, I still absorb that light. Not as much, but I absorb it. So we will still have a response. We're absorbing the light, it'll give us a response. The response here won't be as strong as the response is here, but we'll still have a response. Okay, so if we shine pure 420 nanometer light, okay, something like this, you're going to perceive a purple. Okay, it's purple light. We'll get a fairly large response at this short cone. If I do 450 nanometers in here somewhere, I still detect light, but my response isn't as strong. Okay, so your brain can distinguish these two wavelengths. If it's full, we'll see that. Now, if we have, let's say 70%, if I just tell you that my short wave cone responded at say 70% of its max absorption, there's two places on this curve where that could occur. So we could be looking at this, or we could be looking at this. Just based on this, we can't tell the difference. We can't distinguish different wavelengths. Okay, so individual cones have a peak response. They respond best to a specific wavelength. However, they respond to many wavelengths. Look at this. We go all the way from 450. We have a response all the way up to, say, 600, 650. Okay, so we do have a response over to to light of a broad spectrum of wavelengths. All right, so again, this is just, we have a maximum response here. Any other wavelength, we do still have a response. We are still going to get action potentials going to the brain, right? But our response won't be as strong. All right, and when we talk about color vision, I'm going to come back to this diagram. We'll talk more about the trichromatic theory. All right. One last thing before we go, and I'll schedule another session. This is just a rough overview, kind of showing you the connections of the visual system. So we start with our photoreceptor. That's going to send information to the bipolar cell. The bipolar cell is going to send that information to retinal ganglion cells. The retinal ganglion cells are then going to bundle their axons together to form the optic nerve that leaves each eye. Once we get to the brain, we're going to change direction a little bit, and those axons become the optic tract. So this is eye to brain. And this is basically brain. All sensory information, except for olfaction, goes through the thalamus. Okay, so the lateral geniculate nucleus, I will abbreviate the LGN. This is part of the thalamus. Okay, so the retinal ganglion cells send their axons all the way to the lateral geniculate nucleus. From there, they make some synapses. Information from the lateral geniculate nucleus is going to go on to primary visual cortex. So V1 is the same thing as primary visual cortex, which is the same thing as striate cortex. So you'll hear all of these if you go onto the field. This is where perception starts. From here, we go to many brain regions. So we have parts of the brain that will analyze movement, color, things that will allow you to identify what you see. Okay, so we're here at the ganglion cells. We'll get through the rest in the next lecture that I schedule. Let me stop that recording now.